Dear ladies and gentlemen, colleagues here in audience and also those who are joining us online, it is my honor to start our next parallel session on the scientific activities and projects. First of all, I would like to introduce with my co-chair, Professor Riekstiņa from University of Latvia, and I'm Associated Professor Zanda Daneberga representing Rīga Stradiņš University. Today in our session, we will have 4 young researchers showing their work, efforts and research, which mainly are focusing toward the precision medicine. And as far we have only 4 instead of 5 which were in the program, I guess we will have time to ask questions right after the presentations. And I also would like to encourage people who are watching us online to send in a chat also questions. So we will ask them here in audience. Right, I guess we can start with our first presenter. It's Kārlis Pleiko from the University of Latvia, and his presentation today will be about Aptamers targeting carcinoma cell surface receptors. Please, floor is yours.
Thank you. Thank you for the introduction, and it's my pleasure to be here. Yeah. Okay. I usually start with the introduction since aptamers are not that commonly known targeting reagents. I introduce what they are. And aptamers are short, single-stranded oligonucleotides, so basically short pieces of RNA or DNA that can fold into 3-dimensional structures, and because of these 3-dimensional structures that they form, bind to a specific target protein, either in vitro or in vivo. And on the left side, you can see the secondary structure of aptamer with multiple hairpin structures and one large loop. And on the right side, you can see a protein crystal structure, crystal image of protein bound to 2 identical aptamers on both sides. And this mechanism that nucleic acid binds not due to the strand complementarity, but due to this 3-dimensional structure is unique to aptamers. And mostly, other nucleic acid therapeutics like microRNA or short hairpin RNA binds due to this strand complementarity. And with every targeted therapy that aims to deliver a payload to cancerous site, there are 3 main components. First being the targeting agent itself. In our case, we use aptamers as targeting agents. Because we want to work with emerging therapeutical tools rather than with antibodies that are already established and attracted a lot of funding from pharmaceutical companies. And the other part, the second part is therapeutic agent itself. So we can deliver either chemotherapy, therapeutical drug to the site that we want to, or we can deliver radioactive agent, nanoparticles, other nucleic acid therapeutics like silencing RNA to silence certain genes, and anything else. But we work with the targeting agent part, so to speak, to guide these drugs to place where we want them to be. And the third part is a linker that connects these 2 parts in a one single compound that is delivered to a place where it's needed and exhibits its activity there. And aptamer selection method, CellSelex, this is what we use in our lab, starts with a large randomized oligonucleotide library, and each library contains approximately 10 to the power of 15 unique aptamer sequences. And the selection process starts that we incubate this library with healthy cells because we don't want to target them. We keep the unbound sequences and then apply these unbound sequences to the cancerous cells, the cells that we want to target. And after the incubation, we wash away the unbound cells or weak aptamers or weakly bound aptamers and keep the ones that strongly bind to the cancer cells. Amplify using PCR and repeat the cycle until the library contains mostly only the sequences that bind to cancer cells. And we did exactly that and identified green B1 aptamer as a good binder for triple negative breast cancer cells. And on the upper row, you can see randomized oligonucleotide library binding different concentrations to these triple negative breast cancer cells. And these are mainly just dark images because there's no fluorescent signal coming off when we use imaging flow cytometry. And with green B1, we can see that already at 25 nanomolar concentration, there is a bright fluorescent signal, meaning that the aptamer binds to these cells at very low concentrations. But this doesn't help us much if we know only that the aptamer binds to cells, because we don't know at this point the target protein that it binds to. And if we don't know the target protein, there's a limited amount of applications where we can use it. We don't know the pathways that it can interact with and what effects it might exhibit. So we used proximity labeling method where we conjugate this green B1 aptamer to Horseradish peroxidase. Incubate again with triple negative breast cancer cells and then add tyramide biotin. And this reagent together with hydrogen peroxide creates reactive tyramide species that bind covalently to nearby proteins and target proteins of the aptamer. And then we can use easily just pull down these target proteins and use mass spectrometry proteomics to identify the target. And based on mass spectrometry data, the most promising target was beta-1 integrin. It has already been linked to invasive breast carcinoma and also essential factor for forming vasculogenic mimicry. So tumor blood supply mechanism where tumor cells form blood vessel-like structures themselves. And what was also interesting that green B1 internalized in cell and was trafficked to lysosomes, so we can deliver something inside the cell. And when we measured the affinity against purified beta-1 integrin protein, we saw that the KD value, so affinity value, is 15 nanomolar, so in low nanomolar range. But again, if we know that it binds to target cells, we know the target protein, we still don't know if it works in vivo. There's a lot that can go wrong when we start translating in vitro data from in vivo to in vivo and to test if this green B1 aptamer can still reach the tumor in mice bearing triple negative breast cancer model. We injected both randomized oligonucleotide library and green B1, and after 1 hour circulation time collected the tumor itself and also control organs, so heart, kidney, liver, and lung. And then calculated the amount of green B1 in each of these sites, and also the randomized oligonucleotide library. And we didn't see any differences in control organs, so this is what we want. We don't want to target healthy organs, we don't want to bring chemotherapeutic drugs there. But we saw statistically significant and very convincing accumulation of green B1, more in tumor than we saw with randomized oligonucleotide library. And on the right side, you can also see the whole tumor imaging data with green B1 injected. These are images of whole tumors there. And the bright color is green B1. And in the lower row, you can see randomized oligonucleotide library. And you can see that there's a lot more bright spots in green B1 sample than compared to randomized sample. So we effectively saw that it also works in vivo. And of course, we want to expand on this, and we have several future directions on mind. And one very straightforward way how to move further would be to create a drug conjugate to deliver the chemotherapeutic drug. But we thought about the mechanism where green B1 delivers this chemotherapeutic drug inside the lysosome, and we could use lysosome-sensitive linker to release this chemotherapeutic drug only when it internalizes in the lysosome. And the other approaches are more innovative, targeted protein degradation, and we could create a lysosome targeting chimera, linking together green B1 with another targeting agent, and lead that would bind to protein of interest that we want to degrade. And this complex would be able to degrade these proteins of interest by directing them to the lysosome where they are degraded. Yeah, and that's it for now. And thank you to my colleagues at the University of Latvia and also University of Tartu. And of course, Microtech for supporting me during the PhD studies.
Thank you for your presentation. Any questions from the audience or online? No, so maybe I have one question. So I understand that your research is somewhere in the middle. It's quite far still from clinical practice, but if you think in the future, what would be advantage of this Aptamers compared to the conventional methods used already now for delivering therapeuticals to the cells? Thank you for the question. The other alternative would be antibodies, but antibodies have been already researched as targeting agents for integrin proteins, and the problem is that they have very high molecular weight. The size of classical antibody is around 17 nanometers, and for Aptamers it's 2 nanometers, so there are several orders of magnitude smaller than antibodies and could reach these poorly vascularized tumor sites. Okay, thank you. So sounds really promising. Good luck with your research in the future. Thank you.
And we continue our session with our next speaker, Katrina Daila Neiburga from Rīga Stradiņš University. And the title of the talk is RNA-seq in the molecular diagnosis of primary immunodeficiencies. So the floor is yours.
How do I... Okay.
Okay, in this case, I would like then to suggest to proceed with next speaker, and you sort out the location of your presentation. Would that be okay? Yeah. Okay. Thanks very much. So then I will invite the next speaker. That is Ērika Bitiņa-Barlote from Rīga Stradiņš University, Latvia, and Ērika will give a talk on biomarkers for early diagnosis of bladder tumor. Yes, you're welcome to start.
So hello, dear colleagues. My name is Ērika Bitiņa-Barlote, and I'm a PhD student. And today I want to tell you about biomarkers for early diagnosis of bladder tumor. So, as we see, there are 3 high points which we need to proceed that biomarkers should be with high sensitivity and specificity, easy to use, and of course the complexity and the course is also matters. So, and what now is available? The golden standard methods, what is now used in neurology, is cytoscopy and urine cytology, but they are with limited sensitivity. Because not in any case we can see the tumor by cytoscopy, and cytology in low-grade tumor is only 50% high sensitivity. So, and they are also used for surveillance. So, of course, there are also new biomarkers available, but they're highly sensitivity, but they are not prior the cytology what we are already using now. So there is a need to find new biomarkers which will be more specific and sensitive in non-invasive disease. Because it really matters, because in a high-grade disease, which is invasive, there are really low surveillance numbers. So, as we see, we find out exosomal urinary microRNA, and as we see, exosomes are small vesicles containing unique RNA and protein cargo. Here we see this exosome, which contains also microRNA. And microRNA are small non-coding RNAs that repress gene expression through matrix RNAs. So, and what is really good with these exosomal microRNAs? They are readily available, easy to collect in large quantities because the urine is in large amount. It is really non-invasive source, for example, compared with blood tests. We have direct tumor contact. It incorporated into exosomes, and that is why it is more stable than their cellular counterparts. Because as we see, exosomal microRNA is really extremely stable. Also, when we through it from the -80 to -20, and also in the 4 degrees, there are also studies about that. And here we see, we analyze the 755 microRNAs by real-time PCR, and we find out 96 differently expressed microRNAs in low-grade tumor and 78 in high-grade tumor. So, and this microRNAs was with at least 2-fold change, and of course p-value was less than 0.05. So we isolated it by microRNA exosome kit, and mature microRNA profiling was done by using microRNA microRNA PCR system, and then real-time PCR was done on VIA7. So here we see our results. Of course, it is a little bit hidden because in the future there will be patent. So here we see the changes of level of TP53 targeting exosomal microRNA, and here we see the control group, low-grade group, and high-grade group. And as we see, this expression is higher in high-grade group. And it also can predict that maybe this disease is more aggressive and that the expression is higher. That is why in high-grade disease. Here we see the changes of level of 10 targeting microRNAs. Sorry for a little bit. And also there is a well-known MIR21. So it is also high expression also in other tumors, also in inflammation, so it is not so specific, but as we see, it is elevated. Also, we done we find target gens in low-grade disease. There was 1,425 predicted target gens, and from then we found out these 20 hub gens, which is in this highly expressed microRNAs, low-grade group. And here we see the high-grade group, top 20 hub gens, and as we see, the most central hub gene in all 2 groups is TP53, which is already known tumor suppressor. So, and in conclusion, I want to say that exosomal microRNAs is a potentially good diagnostic biomarker. They can differentiate between high-grade and low-grade bladder cancer. The main differentially expressed microRNAs are located under TP53 regulation. And the possible changes in microRNAs during treatment could be used as a predictive biomarker, but this is in the future. So thank you for the attention. Maybe there are some questions.
Thank you for your presentation. Questions from the audience, please, or from the chat? Okay, then I take the advantage of asking a question. Really interesting talk, and you study exosomes, and exosomes belong to extracellular vesicles. And how did you prove that these are exosomes that you isolate the microRNA from?
Of course, it is a good question because I also read that in some studies watch if there is already exosomes, but we have this kit special to isolate exosomes. But of course, we don't check if there really are, but our results prove that they are connected also with bladder cancer because we find out this predicted target genes and target genes, and they are all already known to be connected with bladder cancer.
Okay, thanks. Yeah, really promising work. Good luck in the future. And then proceed with next speaker.
Yeah, so Katrina Daila, you would be ready now to give your presentation. So once again, I will introduce with you. So Katrina Daila Neiburga from Rīga Stradiņš University will talk about RNA sequencing in molecular diagnosis of primary immunodeficiencies. So floor is yours.
Thank you. In a bit in a contrast to previous talks, I will be talking more about what I'm planning to do, as this is what I'm doing in my PhD thesis, which only recently just started. First about primary immunodeficiencies. What are they and why are they important? It's a group of more than 400 rare disorders, also called inborn errors of immunity. And as the name suggests, it's genetic defects that lead to dysregulated immune response, which can lead to susceptibility to different infections, autoimmunity, and also increased risk of malignancies. Besides clinical diagnosis, it's also important to identify causal variants, because first of all, it can confirm clinical diagnosis. It can lead to choosing treatments, as in part of those disorders, there is a specific symptomatic treatment. It can help with early interventions and also with genetic consultations of the family. Currently, the etiology is well, not currently, it is complex, because there are multiple layers from genome, epigenome, immunological factors, and it cannot be established in most of the patients, mostly because we have limited knowledge about the genotype and phenotype associations for rare variants. As I mentioned, there is increased risk for malignancies due to defects that are possibly responsible for DNA repair or susceptibility to oncogenic infections. But what's interesting is that PIDs exhibit unique malignancy profiles. For example, for a lymphoma, PID patients have up to 13-fold increase in risk compared to general population. And these malignancy profiles differ from patient to patient depending on their exact diagnosis, which why there is a hypothesis that possibly for some of these malignancies, they could be caused by molecular pathways being overstimulated as compensation for these immune defects. Therefore, understanding the etiology of the immunodeficiencies could also help then understand the etiology of malignancies. And as well, besides the diagnostics, there are challenges in treatment because there is increased risk of side effects during some treatments and also risk of worsening immune defects in case of, for example, immunotherapy. Currently, the gold standard to do molecular diagnostics would be whole genome or exome sequencing. However, it can currently uncover only up to 16%. And why is that? It could be a possibility that there are genes that are not yet associated with primary immunodeficiencies, as new variants are reported each year, increasingly. And the second one is that the non-coding variants are underestimated, as 85% of all the associated variants are in the coding genome, and which we know constitutes only 2% of the entire genome. And whole genome sequencing can catch these non-coding variants, but there's a problem with prioritization if we don't have functional information in, that can support it. In other rare diseases, possibility to include RNA-seq as additional layer has been explored in different muscle dystrophies and others. There have been several studies, and the two main takeaways that they have is, first of all, there is increase in diagnostic rate depending on the study, the disease, the group. It can be up to 36% currently. Why not more? Possibly because there are events that don't really show up in the transcriptome. But the problem with this is that there needs to be disease-specific tissue. For primary immunodeficiencies, this problem is solved as leukocytes are affected, and so blood is clinically available tissue and also disease-specific. But that introduces the problem that blood is a mix of different cell types, and there might be differences in this mixture due to disease-specific reasons, but it also could be disease unrelated, for example, because of age and other factors. So that could influence this application and the expected diagnosis rate. So the first then research questions are how and if this can be implemented, and what to do with possible confounding factors. There are multiple molecular events in the transcriptome that previous studies have looked at. There are splicing and expression outliers that have, because those events have been linked to pathogenic molecular events. Also allele-specific expression, because that can detect haploinsufficiency. And other researchers on top of this have looked also at gene fusion and structural variants. There are different tools for doing this, and that presents one of the challenges of my work, to distinguish what is useful, and as there is no current benchmarking for these methods. As to do with heterogeneity of blood, best to research that would be single-cell RNA sequencing, but down the line in diagnostics that wouldn't be very viable to use, as it is expensive. And therefore, if it would be possible to deconvolute bulk RNA-seq data using digital cytometry, for which there are, again, already people have done it, especially in blood cells, then if this can be incorporated and maybe improve the uncertainty of this RNA-seq analysis. So yeah, all of this then would lead to discovering potential disease-causing variants, which then would need to be validated experimentally, and guide discovery of functional effects of genomic variants. And the two main purposes for doing all of this is to clarify diagnosis for patients that can't be done with the genome currently, for those patients, and establish this as a framework that can be used in diagnostics with reliability and accuracy that is needed for diagnostics. And then currently this all is in what I would call a validation stage. We have a group of patients for which we know genetic variants that are responsible for their conditions, and therefore we can compare to what we see in the transcriptome and see what we can or maybe what we can't see. And I would like to show some examples. One is this. There's a patient with loss of function variant in BTK gene. And in expression data, we very clearly see that there's a decreased expression for this gene for this patient. And it's been reported that this variant leads to skipping exon 5 in this particular gene. And here we can see the top one is the patient, and the bottom two are other patients for comparison, and we indeed can see that there is an exon skipped. Another case, another patient with a loss of function variant in a different gene, and we would expect to see something similar with the decreased expression. But curiously, in the expression data, we don't really see anything. And if we look at the expression of this gene among our samples, then this case falls somewhere in the middle, which, of course, makes you ask questions. But biologically, this could also be explained by this transcript escaping nonsense-mediated decay, and then be an example of something that we can't really see in the transcriptome, and that maybe needs additional multi-omics layers to then see this change. There are also, besides these so-called controls that we can look at, there are already some intriguing insights, and I want to share one. A patient with unclear primary immunodeficiency diagnosis who has non-Hodgkin lymphoma. What the RNA-seq data show is that in this particular gene, they have a splicing aberration. And there is, for some of the RNA, there's an exon that is skipped. And what is interesting is that splicing defects in this gene is associated with another disorder that has clinical features that often overlap with primary immune deficiencies, and in the literature it's talked about that it's hard to distinguish between two oftentimes. So this is an interesting case to look into then further. So in summary, currently primary immunodeficiencies are underdiagnosed, and genome sequencing is not enough. And what I'm exploring is adding RNA-seq as additional layer to clarify, seeing what sort of problems arise in trying to implement it and how to correct for them, with the goal then to increase the diagnostic yield and establish this for diagnostic use. I would like to thank my 4 great supervisors for this, both from University of Tartu and Rīga Stradiņš University, as well Microtech for sponsoring this, for funding this, and colleagues from ARSU and Children's Clinical University Hospital for doing the lab work so I can have bioinformatics data to work with. Yeah, thank you for your attention. If there are any questions.
Thank you.
Yeah. Oh. Thank you very much. So any questions from the audience or from the chat? So maybe I can use the situation and ask a question. So now it's a great job to look into the primary immunodeficiencies and the genetics and the diagnostics. But as you said in the beginning, there is quite a lot of unknown genetic background, but you can't really discover. So do you have any information approximately what part out of this unknown reasons could be dedicated to the epigenetic regulation? Because we know that the immune system has very specific epigenetic regulation and, for example, monoallelic expression like somatic rearrangements. Is that mechanism somehow covered also in analysis of the transcriptome?
Not currently, not by me, but there are plans within these projects to also look at epigenetic factors to see. So currently it's very hard for me to say like what percentage could be responsible for like any of it. But yeah, it's also thought about. So, yeah.
Okay.
Maybe not necessarily by me.
Thank you. Yeah, any question?
Yeah, I would also like to ask, it's very intriguing indeed, you are going into unknown, but is there any known RNA profile of these primary immunodeficiencies, or you just pick up genes one by one?
Well, not really, and yes, I mostly look at the genes who show these specific like aberrations, so to say, and then compare them whether they are immune linked or they have any sort of reason behind them to explore them further. Yeah.
Okay, thank you. And we proceed. If there are no more questions, then we continue with our next and I believe last speaker for the session. Yeah, and I would like to invite Nora Kriķe from Rīga Stradiņš University, Latvia, to present her talk on copy number variation and BRCA1/2 inactivation in breast cancer tissues. So the floor is yours.
Yeah. Good day, audience. It's a pleasure to be here and a part of this forum. I'll present a really smart part of the research that I've started doing since I was a med student, because now I'm kind of a medical doctor and also trying to do this as my PhD research. Briefly about the institute where the work is done. So I started my research work about 4 years ago as a cancer and genetics enthusiast, and one of the main research projects is about breast cancer research, but it's not limited only to that. We have biological material collection that consists from breast cancer and healthy breast tissue samples from more than 1,700 patients by now. From the breast cancer center in Kārlis Pleiko Clinical University Hospital. And how it all started? Well, we have a lot of samples, a lot of information from previous patients, and we started the idea that we wanted to kind of profile, let's say, look at copy number variations in the samples that we have and what samples come to us from the breast disease center. And my research is mainly focused on somatic events in breast cancer tissues. So the group that I'll present my results now are women with breast cancer. They have not previously received neoadjuvant chemotherapy, and they have undergone surgery in the Clinical University Hospital. I gather these samples that we have, and I did copy number variation and methylation status analysis performed using multiplex ligation-dependent probe amplification, or MLPA for short, using 2 different probe mixes. For a total of 47 genes, including 21 oncogenes and 26 tumor suppressor genes. That also includes BRCA1 and 2, and also RBB2 or HER2. And this technique is PCR-based, so cheaper than any NGS sequencing analysis. So we stratified the sample group into 3 different groups. The first group is samples with BRCA1 and 2 germline inactivation or hereditary cancers. The second group is where we discovered BRCA1 or 2 inactivation, somatic inactivation in the samples by either deletion or methylation. And the third group is the sporadic cancers that we did not discover any BRCA1 or 2 changes, somatic changes, and also they didn't have germline changes, of course. This is the kind of total data that we have so far. As you can see here, this blank space, each row represents an individual sample, and here below you can see the different genes, the copy number variations we've done for. This blank space, we discovered that more than 30% of genes don't contain any somatic copy number variations, or no variations at all. In total, here are represented 357 samples. Here they're stratified by the corresponding groups, so 10 samples in the first group, 46 in the second group, and 301 in the sporadic third group. The most commonly deleted tumor suppressors that we discovered, you can see here, is CDH1 and 27%, and then 9.5% each BRCA1 and VHL. And as you can see here, this cluster, this is in the somatic BRCA1 and 2 inactivated group. Then, most commonly amplified oncogenes, so 22% MYC gene, this row here, and 16% the MTDH gene. What we did also was we just counted all the how many aberrations have happened there. So I don't know if deletion happened in 3 genes, then so it corresponds to 3 changes in copy number variation. And if we compare these groups 3, then we can see here that there's a statistically significant difference between the somatically inactivated BRCA1 and 2 group and the BRCA kind of negative, where they don't have any changes in copy number variation or methylation of these genes. This is just an example from one sample with somatic BRCA1 deletion that you can see here, the BRCA1. And this sample contains a whole variety of aberrations, including FGFR deletions, MYC amplifications, MTDH, CDH1 deletions, RBB or HER amplification, VHL also. Yeah. So this is only a small part from the work I plan to do in general. So for now, we've included also NGS sequencing for whole BRCA1 and BRCA1 or BRCA1, BRCA2 genes, and also panel sequencing for cancer hotspots for detection of smaller or point mutations, because of course, let's say TP53 is also commonly mutated in breast cancer, and maybe these mutations are just a byproduct of it being inactivated. Yeah. Also, about half of the samples I use, we have clinical data gathered. I do it myself, so we'll also add that to the research later on. Yeah, and maybe might some of these variations also work as new prognostic markers. This is from a talk I did about 2 years ago. Here is shown overall survival with for patients that had APC methylated in their samples. Here, CDH1 deletion and overall survival. We can see a tendency, but 2 years ago, when doing this analysis, it was not statistically significant. Yeah. So, thank you for your attention. Any questions?
Thank you for your great talk. And yeah, I'd like to invite audience or chat for questions. Then I would like to ask a question myself. I guess you mentioned that this is your future plan, that you will try to correlate these findings with clinical data, like response to treatment. But so far, maybe you can comment, maybe there are data from literature that have studied this. Is there any correlation with treatment response? Or?
I forgot to mention an important note. This research is entirely retrospective. We kind of don't follow up with extra kind of samples, what changes have been, somatic changes have happened during the treatment, because yeah, the samples are taken from post-operative breast tissue, so we don't biopsy women who haven't undergone surgery. About literature, I compared also the most commonly deleted or duplicated or aberrations in copy number variation, and they kind of correspond also to our samples that are reported in the literature more commonly. About therapeutic approaches, I haven't looked into that so far. But in the future, yeah, we have to consider different like these histological subtypes, hormone positive, triple negative, HER positive. Of course, stratify, look, maybe there are some differences there because breast cancer is very complex, but this started as a general database gathering about what are we dealing with in the clinic.
Thank you. More questions? No? If no, then I give the word to the Professor Daneberga.
Yeah, thank you. As far we don't have a last presentation, so we still have some chances to ask questions to presenters, if any arise for the previous presentations. I guess there is nothing in the chat either. Okay, as like a conclusion, maybe I can just say a few words that this kind of young researchers who are working in the research field and doing some research in oncology, I guess that's also important part of the precision medicine, because we know that this kind of new discoveries help us to come closer to each single case of the patient. So in general, I would like to wish good luck with your research, which is really important work what you are doing. And yeah, I guess that's it for today's session. So we finished a bit earlier, but from 2 o'clock, where is the coffee break and networking? So we can do networking now, but coffee break at 2 o'clock. Thank you very much for your attention.